Hello, and welcome to part 6 of our innate immunology lecture. In this section, we're going to be talking about a chemical defense, another second-line defense in innate immunity, known as the complement system. And complement is a set of proteins that are produced by the liver, and they are circulating around in our circulatory system, so they're found in blood. In a normal state, they are inactive proteins. They are part of the innate immune response, and they do have some effector functions, but when we are in a healthy state, they're inactivated. They're not doing anything. But in order for these cells, for these proteins, to be activated, they will go through one of three different classical, what we call activation pathways. We have classical activation, alternative activation, and the lectin pathway. All three of these activation pathways will result in something called C3 convertase. And C3 convertase is a protein complex, or C3 protein complex. This convertase activity then follows through another cascade. Think about the clotting cascade that you may have learned about in anatomy. So the complement cascade is a series of proteins that all activate each other like a row of dominoes. And one will activate the next, will activate the next. And the results or effects of the activation of complement is increase in inflammation, another process known opsonization, which is enhancing or enabling phagocytosis. And the next is the membrane attack complex formation, which results in cell lysis of the bacteria cells, not host cells. So we have classical complement, which activates C3, the C3 protein, and then we have the alternate complement activation pathway, which starts with C5. So in complement activation, we have these three pathways. Here we have classical activation. And one thing to notice in classical activation is C1, the very first complement protein, must bind to antibodies. This means that the classical pathway can only take place in secondary immune responses. The very first time that you are exposed to a pathogen, the classical pathway will not work. And this is because you don't have any antibodies. So until you have antibodies, your classical pathway will not work in terms of activation. Once you do have antibodies, two antibodies will bind to the antigen or microorganism, and C1 will bind the branches between the two. So this is known as cross-linking. And when C1 cross-links, it will attract C2 and C4. C2 will be activated, C4 will be activated, and this will cause these two proteins to split. When they split, they become parts A and B. C2b and C4a are what we call anaphylatoxins, and these anaphylatoxins will enhance inflammation. So they go out and they are part of the inflammatory process. C2a and C4b will come together to form C3 convertase, and that's what we're looking at right here. So C2a and C4b come together, we get activation of C3 convertase, and this then sets off the complement cascade. In the alternative pathway, we have direct activation of C3 convertase itself directly, and this direct activation results in the same complement cascade being activated. So in this one, we do not need any kind of antibodies or intermediaries. Factors B, P, and D, these are usually found on the microorganisms themselves. They're part of that lipocarbohydrate complex, and so we get direct activation of C3 immediately. So this will be a very rapid response in primary immune reactions. Then we have the lectin pathway, and in the lectin pathway, the liver produces some additional proteins known as lectins, and these lectins recognize particular molecular lipocarbohydrates that contain mannose. Mannose is a sugar used in a lot of the carbohydrate signaling molecules found on the surface of bacteria. So specific mannose patterns can be recognized by one of the lectins within the class of lectins, and when lectin binds to that mannose pattern, when it finds its mannose pattern, this will directly activate C2 and 4, And we then have C2A, C4B, and the formation of C3 convertase. So all three of these are three different ways to activate the same process, which is that C3 convertase cascade. And the difference is really that, take a look at the classical pathway. This has the most components. And in the classical pathway, we have antibodies, secondary response, C1 protein, and then activation of 2 and 4, and then C3. This, my alternative pathway, we just totally skip the whole, this whole process, steps 1 and 2 here. This gets completely skipped. And then here in the lectin pathway, we simply are replacing the antibody C1 complex with the lectin mannose complex. But the activation pathway, step 2 here, is the same. So now that we have looked at the three different ways that complement can be activated, we take a look at the actual complement cascade itself. So here we have our three pathways, alternative, lectin, and classical. And no matter how it's activated, any of these pathways, we get formation of C3 convertase. This complex here, once activated, causes the protein C3 to divide, and C3 splits into C3A and C3B. C3A is a strong anaphylatoxin and also plays a role in inducing the inflammatory response. So it increases inflammation. In C3B binds to the surface of bacterial cells, and so the bacterial cell will get coated with a whole bunch of C3B proteins. And these C3Bs are ligands for receptors on many of our phagocytic cells, and this is how our phagocytic cells carry out receptor-mediated endocytosis. And this is a process that phagocytic cells use in order to ensure not only phagocytosis of the microorganism, but actual destruction of it as well. But C3b has a second job. You notice there are two arrows here, and the second job is to activate C5. C5 will then split into C5a and C5b. C5a is another very potent anaphylatoxin, and this can cause a very strong immune response. C5b will then attach to 6, 7, 8, and 9, forming the membrane attack complex. And this causes the lysis of foreign cells. That works kind of like a hole punch. And so these C9s will congregate in this big tube-like structure and literally punch a hole across the outer lipid layer of gram-negative cells. When we punch a hole through that outer lipid layer, all of those protons, right, all of those protons in that periplasmic space will leak out. We start releasing all of these protons. The bacteria can no longer utilize their flagella. They can't power it, and they, of course, can no longer power ATP production. Now, the activation pathway leads to C3 convertase formation, activation of this entire pathway here. It is highly regulated. C3b will become inactivated if it tries to bind to a host cell, because it can't form a convertase action. So notice C3b here, if it tries to bind to a host cell, to one of your own cells, it will be inactivated and can't function. But if it's bound to a bacterial cell, it is exposed on a side that is complementary to receptors found on phagocytic cells and initiate that phagocytic process. So opsonization, it's not that these cells would not necessarily phagocytize the bacterial cell, but by coating it with C3b, they can grab onto it more quickly. Think of C3b as kind of making it sticky. A way that I like to remember which proteins do what, C3a, C5a, inflammation, C3b, C5b, bind. Okay, so the B's bind, the A's are part of inflammation. And that's just a fun little way to kind of remember which proteins in complement do what.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2A and C2B, and C4 into C4A and C4B. C2B and C4B combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3A and C3B. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3A and C3b are constantly being formed from C3, but without activation, they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3A and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3A is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3A and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properdin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5a and C5b. C5a enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components, including C6, C7, C8, and C9, to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
So you can see the three effector functions of complement are extremely important in our innate system. Without any kind of adaptation, antibody production, specialization, or presentation, our innate immune system can play a really important role in protection and getting rid of these organisms or pathogens very quickly. So just to review real quick on complement, you want to make sure you know what complement is, what it's made up of, and where in the body it is actually produced. What are the three different activation pathways? You should know what C1 does, what C2 does, what is C3 convertase. You saw in that video there's a couple of different forms of it. How does C3b cause activation of its own complete C3 convertase? So C3b turns around and actually creates a new convertase. Which pathway requires the adaptive immune system? How does complement create a positive feedback loop for continued activation? How does it keep reactivating itself? What are the effector functions? Sorry for the typo, this should be the other way around. What are the effector functions of complement, and what proteins are responsible for those effector functions? So which complement protein is responsible for which effector function? So that's the end of part 6 in your introduction to the complement cascade. And this is a really important part of the innate immune system. You will run into this again in your pharmacology classes. They do talk a little bit about complement in that sense, and in process 1 in the nursing program. So I hope you enjoyed this lecture. I will see you all in part 7.